We'll start in just 1 minute, but I'll remind the audience that next week is the last session of this academic year. It will start up again in September. So I certainly welcome suggestions from our audience as to topics they'd like to see on the teaching series next year. So go ahead and send me an email about any thoughts or suggestions. Having said that, and it's 7 o'clock, Dr. Choy at Genentech kindly going to be our speaker this morning on lung dysbiosis and a host molecular profile of activated innate immune function as an endotype of COPD. That's a mouthful, but I'm sure he'll lead us through that. So thanks again for joining us.
Well, thank you for the introduction. And thank you for the organizers for giving me the opportunity to share our work over here at Genentech. So once again, I'm Dave Choy. I'm a distinguished computational researcher at Genentech. The focus of my lab is the study of muco-obstructive lung diseases, including asthma and COPD, so chronic obstructive pulmonary disease. And I'm really excited to share some of our recent work today in endotype and phenotyping of COPD patients. So as in terms of my disclosure, I'm an employee of Genentech and I hold Roche group stocks and options. So why are we interested in endotypes? Well, first of all, I think if we look at the example from asthma that was borne out nearly a decade ago, there's really an appreciation that asthma, the diagnosis of asthma, is really an umbrella term for a series of phenotypes, subphenotypes that may, that may underlie the disease. And these subphenotypes are potentially endotypes. And what endotypes are molecularly defined phenotypes. and this has implications with respect to personalized therapies and targeted therapies. The example of this is that over a decade ago, Prescott Woodruff at UCSF, in collaboration with Genentech, we had defined a molecular subphenotype of asthma, as you can see here, based on this heat map. Genes that are inducible by type 2 cytokines which were induced to various degrees in asthma patients, and it overlapped with healthy controls. Linked to this endotype or phenotype were peripheral biomarkers such as blood eosinophils that subjects who had high expression of these Th2 expressing genes had high levels of blood eosinophils. Now today is really kind of borne out with targeted therapies to targets related to the type 2 pathway seem to have meaningful clinical benefit in subjects with type 2 high asthma as defined by high levels of blood eosinophils and pheno. And really, I think this represents an example that we could potentially learn from with respect to other chronic lung diseases that we're studying. So I would just probably would just remind everyone about chronic obstructive pulmonary disease, which is a distinct lung disease from asthma. And really the focus on COPD was really due to that substantial unmet medical need. So a high risk factor for the development of COPD is exposure to cigarette smoking or biomass fuel exposures. Currently, it's the third leading cause of death. And it really highlights that we need to do more in the context of COPD. As in terms of the standard of care therapies, they're mainly bronchodilators, as well as anti-inflammatories that might be familiar to you in the context of other lung diseases. COPD is a very heterogeneous disease. And as in terms of the recognized histopathology that's been described for COPD is really as in terms of this airway disease that might underlie chronic bronchitis, which is associated with remodeling of the airways, pathologic mucus, inflammation, and dysbiosis. What can co-occur in individual patients is parenchymal disease, where it's really a loss of alveoli, so the final gas exchanging units of the lung. As in terms of clinical presentation, some of the things that we're trying to address in the context of COPD are acute exacerbations of disease. So these are sudden worsening, clinical worsening, necessitating intensification of healthcare utilization. One of the issues about acute exacerbations is that we think that their etiology is heterogeneous with respect to normally innocuous triggers and a maladaptive host immune response. A long-term goal in this disease area is to either slow, halt, or reverse disease progression. And this is kind of a long-standing goal, but really kind of the focus of my presentation is an understanding of exacerbations in the context of COPD. So hopefully what I will try to describe today is really an understanding of potentially an axis in COPD where we're linking potential the molecular phenotypes with pathophysiology and endpoints in the context of COPD. And the way that we've done this is human phenotyping. And in this case, in a study of lung molecular profiling, specifically we did a multi-omics analysis, we're able to show a correlation of activated innate immune cytokine signaling with neutrophilic inflammation in the lung and bacterial lung dysbiosis. And this was a feature of subjects who are frequent exacerbators of COPD. Next, we were able to link it in another mechanistic study. In this context, in our evaluation of a COPD experimental rhinovirus challenge study, we had identified that this phenotype is associated with a reduced ability to respond to rhinovirus with respect to the ability to induce interferon regulated genes, suggesting that this is a phenotype that is susceptible to rhinovirus with a lack of ability for antiviral responses. And then finally, we think this phenotype may be an endotype in our study of an experimental medicine, astegolimab, which is it's antagonistic antibody for ST2, the receptor for IL-33. We had identified that this phenotype is associated with enhanced response to anti-ST2, suggesting that this is a targetable pathway and hence an endotype. And really the way that we have arrived at these conclusions is really based on the convergence of the answers to these three questions that we asked in our respective studies. The first is, what is the host clinical and molecular phenotype associated with lung dysbiosis? So the reason why we asked this is hopefully is based on the motivators on this slide. First of all, if I would refer you to this study from Chris Breitling at the University of Leicester, what they had done is that they had followed COPD patients and asthma patients, and at the time of acute exacerbations had collected sputum. And really their goal was to deeply phenotype this biosample from the lung to better understand the heterogeneity of asthma and COPD exacerbations. What they had done in the study is that from the sputum, they had measured a panel of inflammatory cytokines and chemokines. And what you see here is after factor and cluster analysis, you see the samples projected on linear discriminant analysis scores. And what you can see here is that they overlaid the clusters on the basis of the inflammatory phenotype in the sputum, as well as the microbial phenotype. So first, hopefully what you can appreciate is that there is heterogeneity with respect to the molecular phenotype of exacerbation. And what you can see here, number one, cluster one had been identified really driven by the upregulation of innate cytokines, such as TNF-alpha and IL-1 beta. The inflammatory properties of these particular samples at the time of exacerbation is that there was a high degree of or elevated neutrophilic inflammation. And as in terms of the microbial profile, there was a high prevalence of bacterial culture positivity of the samples. If we take a look at cluster 2, which was really driven by an upregulation of T2 cytokines, such as IL-5 and IL-4 and IL-13, we can see that the inflammatory profile is eosinophilic. So really it's considered to be an allergic exacerbation. And then finally, we looked at cluster 3. This was a viral-associated exacerbation. The inflammatory profile was driven by interferon gamma-inducible genes, such as CXCL10, and there was an increased prevalence of viral positivity based on a PCR panel. So based on subsequent work that we had done, and we had published on last year, is that we had shown that this exacerbation phenotype is really associated with a phenotype of dysbiosis during a stable state of disease. And from the original study, what they had originally identified is that these subjects also had elevated IL-1 beta, even at a stable state, suggesting that we can phenotype subjects at a stable state to predict the type of exacerbation that they're going to have in the future. So with this in mind, with that lung dysbiosis being a potential predictor of a future bacterial exacerbation, we then asked the question about what was the extended phenotype, both from the host and from a clinical and a molecular perspective. So to do this, we studied COPD patients and as well as healthy volunteers. And this is a description of their demographics. And there's a few points I want to mention on this study that I'm about to present to you. That it was a relatively large study where we included up to 153 COPD patients and to a lesser degree, 23 healthy controls. It was quite exemplary in the fact that in this particular cohort, nearly all of the COPD subjects were former or non-smoking subjects. And the reason why I say it's notable is that we realized that there's a confounding effect of current smoking for molecular profiling of the airways. And I also wanted to mention that these subjects were quite severe. So gold stage is a classification of disease severity in COPD. And we can see that the majority of the patients were gold stage 3 and 4. So the majority that you can see here, we see at gold stage 3, there's nearly 17%, and gold stage 4, nearly or greater than 67%. And from the study, from bronchial brushings, what we did is we isolated DNA and RNA from the brushings, and we did bulk RNA sequencing. And from the DNA, we did 16S ribosomal RNA DNA sequencing for an assessment of the bacterial microbiome. So first we asked, what are the clinical associations of alpha diversity? So alpha diversity is a measurement of bacterial community diversity that's irrespective of the organisms in all of the samples. So for instance, a high alpha diversity in 2 different samples are independent of the actual organisms from the 2 different samples. So hopefully what you can see here is that in COPD patients, as reported in the past, is lower in COPD subjects as compared to healthy controls. But that really doesn't tell the whole story. As you can see that there's a high degree of overlap, and there's a minority of the subjects that have extreme low levels of alpha diversity, suggesting that there is a handful of organisms that are dominating the microbial profile. Next, we ask the question, what are other clinical associations with low alpha diversity? So a biomarker that is predictive of future bacterial exacerbations. We can see that subjects having 2 or more exacerbations in the previous year have statistically significant lower alpha diversity as compared to subjects with less than 2 exacerbations in the previous year. And then finally, we asked a question with respect to muco-obstructive pathology or the clinical presentation of it. Subjects were classified on the basis of whether or not they're chronic mucus hypersecretors. And this was really on the basis of patient-reported outcomes that can capture symptomatic cough and phlegm. And surprisingly, what we saw is that alpha diversity was comparable in subjects with CMH versus those without. So really the clinical associations of low alpha diversity was that it's lower in COPD and it's lower in subjects who were frequent exacerbators. Next, we asked the question on a molecular level based on gene expression, what are the correlates with alpha diversity? So there's a lot of information on this slide, and let me walk you through it. based on the correlation analysis that we did, we found that we had a very strong signal. So 850 genes surpassed the threshold of a false discovery rate of less than 0.05. And so what you see here is a tabulation of gene set enrichment analysis. It is a way that we can systematically look at the associations of certain gene sets with respect to associations with alpha diversity. And what we saw here were 198 gene sets from the Go term collection that had an FDR of lower than 0.05. And it's significantly was associated with different aspects of innate immune function, specifically IL-1, IL-6, and NF-kappa B signaling and epithelial differentiation. And I just want to dwell for a second on the volcano plot. And the way that we interpret this is that genes on the left side are relatively upregulated in subjects with lower alpha diversity. So subjects who, i.e., have lung dysbiosis, these genes are upregulated in those subjects. And what we can see here, which is really kind of borne out or underlies the GSEA analyses, is we see an upregulation of IL-1 family genes, such as IL-1 beta, IL-1 alpha, and IL-1 receptor antagonists, and also as well as IL-6 signaling that we can see IL-6 itself, as well as OSM, which is another IL-6 related gene. Also, we see an upregulation of genes associated with neutrophilic inflammation. We see the receptors, such as CXCR1, and as well as the ligands that recruit neutrophils, such as CXCL2. So to summarize this slide, as in terms of what we see correlated with lung dysbiosis is an upregulation of innate cytokines, neutrophilic inflammation, and epithelial differentiation. So going into a little bit more about GSEA, so underlying the analysis that you're seeing here is that it takes a particular set, a gene set, and systematically evaluates it based on the association of underlying genes. And from what you can see here, it evaluates whether or not there's a systematic shift as in terms of the association, as you can see here, using the moderated T statistic as a metric for association. And one of the ways to deal with gene set enrichment analysis is overlap analysis. And really what the problem that this tries to solve is that many of the gene sets within a particular collection overlap with respect to each other. And to get a better sense of the actual genes that are driving the association with the gene sets, we can compute an overlap index analysis. And the way that it helps is borne out here, is that we can, with the overlap index analysis amongst the significantly associated gene sets, we can do a cluster analysis on the gene sets themselves. And what it does is that it brings together gene sets that were significantly associated together. And hopefully we can see here amongst the most strongly associated gene sets, we can see related genes and gene sets brought together, where we can see that gene sets that are indicative of epithelial differentiation, as well as squamous cell metaplasia, are really borne out here, as well as things related to antimicrobial defense, as you can see here, based on the gene sets, and as well as granulocytic inflammation, and finally NF-kappa B signaling, and as well as innate cytokine signaling, such as specifically as IL-1 and IL-6 families. So we think that these recent results that we have are recapitulated by another recent study in the literature. And specifically, what this group had done is that they had developed an IL-6 trans signaling signature from epithelial cell cultures. And when taking this IL-6 trans signaling signature, what they had done is looked at human data sets of COPD and to assess the heterogeneity of the molecular profile in the lungs. And they had identified some subjects who had this high IL-6 trans-signaling gene signature. And some of the features that they saw here was an upregulation of other innate cytokines, such as IL-1 beta, as well as those related to neutrophilic inflammation as IL-8, suggesting that there's some consistency between our recent data and this published study from 2021. So we think the answer to the question, what is the host clinical and molecular phenotype associated with lung dysbiosis, is that the clinical association is that they're frequent exacerbators, and as in terms of the lung molecular profile of activated innate cytokine expression, granulocytic inflammation, and epithelial differentiation. And unless there's any questions, I'll move on to the next question that we asked. What are the host factors associated with a diminished type 1 interferon response after COPD rhinovirus challenge? So the reason why we asked this is that it's been a long-standing hypothesis that there's impaired antiviral immunity in the context of frequent exacerbators of asthma and COPD subjects. And really, personally, I've been calling it that this is the Seb Johnston hypothesis, as he's been popularizing it in a number of studies for quite a while. And representative of this hypothesis, in this case, he had studied COPD patients who were infrequent or frequent exacerbators. And in this case, this is a molecular assessment of sputum samples. And what you can see here is that frequent exacerbators, they have diminished type 1 interferon levels in their airways as compared to infrequent exacerbators. And this has really been a motivator that another company is actually, as a treatment for exacerbations at the time of exacerbation, is really the treatment with recombinant interferon beta in inhaled fashion, suggestive that this might be a way to restore antiviral immunity in frequent exacerbators of COPD. So a big overarching question is, what underlies this impaired antiviral immunity? And so we thought we had an opportunity to ask this question as a part of our evaluation of the experimental COPD rhinovirus challenge. And the reason why we were conducting the study in the first place is that is a potential clinical model for understanding the activity of interventions in the clinic. But really what this goal was initially was an evaluation of this model. And in this study that I'm going to show to you today was our molecular evaluation of this model with 30 COPD patients. It was a single cohort, non-randomized study, and there was no interventions other than just non-GMP grade RV16. We deeply phenotyped these subjects, both clinically and on a molecular level. This is our study design, where it incorporated a bronchoscopy both prior to rhinovirus challenge and 7 days after, where we were anticipating that this would be at the peak of clinical and molecular changes after rhinovirus challenge. And we collected biomarkers, both non-invasively and those from the airway. And just to summarize what we saw is that we saw a spectrum of responses. We were able to successfully infect the majority of the subjects, so approximately 20 of the 30 participants, and we defined a successful infection by a upper respiratory sample viral titer or seroconversion by the time at the end of the study. And as mentioned, we saw a spectrum of responses, both as in terms of symptoms and with respect to molecular markers that we had measured in the study. So this is a result of us asking the question, what were the molecular changes that occurred based on gene expression in the airway? And as in terms of the design of the study, we did bulk RNA sequencing from bronchial epithelial brushings. We included both the baseline samples and the day 7 uninfected and infected samples so we can properly control for it. So on a single gene basis, based on differential expression analysis, because we had a relatively small study, no gene achieved an FDR less than 0.05. And however, when we did gene set enrichment analysis, which is a much more powerful way to interpret gene expression, we saw that 937 gene sets had an FDR of less than 0.05. And we see the top 30 tabulated here. We see, as we would expect, we saw that some of the strongest signals that we saw were type 2 and type 1 interferon gene sets that were induced with rhinovirus. And we're able to look at the underlying volcano plot. We see interferon gamma inducible genes such as CXL9, 10, and 11 are the most strongly induced, as well as interferon stimulated genes in the airway. So really, this was really telling us that we were able to successfully see some of the strongest signals that we were expecting to see. However, that really isn't the full story. And if we go and look a little bit more closely at the data, and based on what we know from the literature and our own experience, the responses to interferon, type 1 interferons is relatively well understood. And on the basis of a previous study that we had done in bronchial epithelial cells, where we had developed a signature of interferon stimulated genes, and we applied that in asthma, we then took that signature and applied it with respect to experimental rhinovirus challenge of COPD patients. And what you're seeing here is really a summary of the ISG expression in the form of what's called an eigengene. And hopefully what you can see here is that the induction that we saw in infected subjects, but really the question that we had was, why was there such heterogeneity with respect to the induction of these interferon stimulated genes? And really kind of the context for this question, hopefully we can draw back to the original hypothesis that there is impaired antiviral immunity in the context of COPD frequent exacerbators, as well as asthma frequent exacerbators. So next we ask the question, at baseline, what is the phenotype on a molecular level associated with diminished response to rhinovirus with respect to interferon stimulated genes? And this was really the result. So what correlated at baseline with respect to the magnitude of induction of interferon stimulated genes? And once again, because of a relatively small study, no single gene passed an FDR of less than 0.05, but from gene set enrichment analysis, we saw over 1,000 gene sets. So a very strong coordinated signal in this data set. And so what really was drawn out was that there's different aspects of upregulated innate immune function, IL-1, IL-6, NF-kappa B signaling, as well as epithelial differentiation. And hopefully this is familiar based on the original question that we asked, what associates with alpha diversity in the earlier study that I mentioned. So once again, there's a kind of a convergence of answers that we see based on the studies that we had. So really the question is, how do these gene sets overlap with those associated with the previous alpha diversity analysis? And if we take a look in the COPD rhinovirus challenge study, which we called Wildcat, there was over 1,000 gene sets that were significantly associated. And the question about what associated with alpha diversity in the cohort that we had of COPD with the University Medical Center at Groningen, we see a high degree and significant overlap. So there are 73 gene sets that overlap with respect to the question that we asked based on differential expression analysis. And specifically, some of the things I want to call out were the actual gene sets related to the IL-6 family, IL-1 family, NF-kappa B signaling, as well as neutrophilic information, antimicrobial defense, and epithelial differentiation. So based on these analyses, we think that there's a convergence of particular phenotypes that we had seen. So what is a potential mechanistic explanation for this? And I think that based on the rheumatology field, there's an appreciation that in general, NF-kappa B and STAT1 signaling are reciprocally regulated. And this is really kind of borne out initially from observations in the clinic, where subjects with rheumatoid arthritis or juvenile idiopathic arthritis that were treated with anti-TNFs, they had a relatively upregulated level of type 1 interferon after treatment. So there has been subsequent mechanistic studies have been shown that with plasmacytoid dendritic cells, that if you treat them with TNF, they have a diminished interferon response after exposure to virus. So really the hypothesis or potential interpretation is that tonic NF-kappa B signaling in COPD lungs suppresses type 1 interferon responses with respect to rhinovirus challenge. So we think the answer to this particular question, what are the host factors associated with the diminished type 1 interferon response after COPD rhinovirus challenge, is that COPD subjects challenged with RB who elicited the smallest type 1 interferon response were significantly associated with upregulated innate immune cytokine activation, including IL-1, IL-6, and NF-kappa B signaling. So unless there's any other questions, right now, I'll move on to the next question we asked in the next study. And this question is, what are the predictors of COPD clinical responsiveness to anti-ST2, which is an experimental therapy currently in clinical development? It's a receptor, specifically it targets the receptor for IL-33. So the way that we think that IL-33 is involved in disease is that in the disease airway, upon an exposure such a upper respiratory viral infection, which has long been associated with acute exacerbations of COPD, and as well as asthma, it leads to cell death and the release of epithelial alarmins, such as TSLP. And in this context, we think IL-33. Notably, IL-33, as opposed to other IL-1 family members, is not constitutively secreted. In fact, there's a chromatin binding domain in which sequesters it to the nucleus of the cell, and it's not until there's mechanical disruption of the cell that IL-33 is released. A number of cells of the immune compartment express the receptor for IL-33, ST2, including those that we see here. And in general, we think that there are two major categories as in terms of immune responses to IL-33 release and activation. Really what we call the default pathway, that in the context of a normal healthy context, we think that IL-33 amplifies type 2 responses. But also what we think is much more relevant in the context of smoke or infection that actually skews the receptor expression of ST2 to type 1 as well as other innate cells is that there's an upregulation of the responsiveness of these innate and type 1 cells in the context of smoke and infection. So here are some of the results of the phase 2 studies of astegolimab, which is a monoclonal antibody for the ST2 receptor, both in asthma and another recent study in COPD. What we see here as in terms of the y-axis, which is annualized acute exacerbation rate, we see statistically significant reduction of acute exacerbation, and we see clinically significant reductions in COPD, as been recently reported earlier this year. So really our interpretation about this, considering that we think that exacerbations are heterogeneous in nature, is that we think anti-ST2 is effective at reducing some, but not all asthma and COPD exacerbations. And what this also kind of highlights, which is is that potentially is that there might be common treatable traits across these distinct muco-obstructive lung diseases. And really, our question that we have here is that is anti-ST2 more effective for certain types of exacerbations or certain patient subgroups? So what we did in the COPD study that was reported earlier this year, and going into a little bit more details, it was a relatively small study with 81 subjects total, randomized 1-to-1 placebo versus 490 milligrams Q4W, and they were evaluated for almost up to a year. And so what's an important aspect of the inclusion criteria is that all of the subjects were recruited were frequent exacerbators. So they had to have a history of 2 or more exacerbations in the previous year. And I thought that this was really a phenomenal study that was conducted, and it was rich as in terms of biomarker assessments. And some of the results that I'm going to show you are really a summary of what we did from our analyses of the sputum microbiome. And also we did sputum proteomics as well as serum proteomics. And really the question is that do biomarkers indicative of these anticipated pathway activity enrich for anti-ST2 responsive? So what you're seeing here is a summarization of our serum proteomics analysis, asking which biomarkers enrich for treatment responsiveness. And the way to interpret what's called a 4-way plot are that proteins that are farther along this axis, having high levels at baseline, enriches for treatment benefit, and conversely, biomarkers on this end of the spectrum, apologies, enrich for treatment benefit in patients having low levels of that particular biomarkers. And what we saw from this analysis, we definitely saw in enrichment that subjects had biomarkers related to the Th1 pathway, neutrophilic or myeloid-associated proteins enriched for treatment benefit. And we can see the individual plots for the genes here, where we can see IL-18, which notably induces interferon gamma responses in T cells. We can see that subjects at baseline having high levels of these biomarkers enriched for treatment benefit, that they had a greater reduction in acute exacerbations upon treatment. And also what we note is that subjects treated with placebo, subjects having high levels of these biomarkers, they tended to have more exacerbations, suggested that it was correlated or related to underlying disease processes. We can also see some of the genes that I'm calling out here related to neutrophilic inflammation. We see CXCL1, which is associated with the recruitment of neutrophils, as well as GCSF, also known as CSF3, which is involved in neutrophilic maturation and release from the bone marrow. And we see an enrichment of treatment benefit, and to some degree, a prognostic association with on-study exacerbations in the placebo group as well. Next, if we look actually what's going on in the airway. And so in this particular case, we analyze sputum supernatants from this study. And what we saw here, remarkably, we saw that subjects who had relatively high levels of innate cytokines or those related to NF-kappa B signaling enriched for treatment benefit to anti-SD2. Some of these are called out here, but if we actually take a look at some of the actual individual plots, we see remarkably that subjects having relatively higher levels of IL-1 alpha or IL-1 beta at baseline actually enriched for treatment benefit for anti-SD2. Notably, while hopefully anti-SD2 continues on in clinical development and hopefully becomes a medicine that's important for patients in the future, we see that exacerbations still, they don't go to zero. So we think that we still have more work to do, but we think that this data highlights other co-expressed mediators in the context of these subjects who potentially may be motivations for drug discovery in the future, perhaps related to the IL-6 pathway, which is also a biomarker that enriches for treatment benefit to anti-SD2. And then finally, closing the loop with respect to our original question, we asked, does the lung microbiome, specifically alpha diversity, which predicts future bacterial exacerbations, does that enrich for treatment benefit? And what you're looking here is a plot where we stratified the patients on the basis of sputum alpha diversity at baseline at the median. And the top plots indicate the placebo-corrected exacerbation rate reduction. So going up is an increased efficacy. The bottom plots are the individual treatment arms, annualized exacerbation rates. And what we can see here is subjects having lower alpha diversity, so i.e. there's that they have lung dysbiosis. Those are the subjects that have greater treatment benefit for anti-SD2. And also something that hopefully that you may notice is that subjects with lower alpha diversity, the placebo-treated subjects, they had greater exacerbations on study in this particular case, suggesting that collectively that we see that this phenotype of associated low alpha diversity, upregulated innate cytokines, they tend to be responsive to anti-SD2, suggesting that this phenotype might be an endotype. So as in terms of what is the relationship between this phenotype with IL-33, since IL-33 has long been associated with type 2 inflammation, and this appears to be a different molecular context. Well, Alison Humbles and her co-authors have reported this study a number of years ago, where they had shown that exposure to smoke shifts SD2 expression on inflammatory cells. And hopefully what you can see here, after smoke exposure, ILC2s actually downregulate ST2, the receptor for IL-33, and we see an increase in ST2, the receptor, expression on NK cells, macrophages, and as well as T cells. And What she has shown here is that this affected their functionality as well. If we take a look at lung ILC2s, after they're exposed to IL-33, we see that smoke-exposed ILC2s has a diminished IL-13 response, so as in terms of their functionality. So conversely, NK cells exposed to smoke, they have enhanced interferon gamma responses to IL-33, both after 4 days and after 10 days, suggesting that smoke shifts the responsiveness of inflammatory cells to IL-33. And we think that this may be a relevant mechanism that we're seeing in the context of COPD. So as in terms of what we think the answer to our final question, what are predictors of COPD clinical responsiveness to anti-ISG2? We think that the answer is a peripheral and lung proteomic profile of activated innate cytokine expression, granulocytic inflammation, and lung dysbiosis. So putting it all together, we think we have a model that looks like this. We think that in some subjects with COPD, that there's an upregulation of innate cytokines, such as IL-1 and of the IL-6 family, and it correlates with neutrophilic inflammation and lung dysbiosis, and this is a feature of frequent exacerbators. We think that this is an endotype because when we block the IL-33 pathway with anti-ISG2, we see that this is also linked to improved benefit as in terms of acute exacerbation rate reduction here. We think that this phenotype is also associated mechanistically with impaired antiviral immunity, specifically a type 1 interferon response to rhinovirus exposure. So we think that potentially clinically we can enrich for these particular subjects by enriching for subjects with by exacerbation history. And potentially, hopefully that this is an inspiration for new therapies in the context of COPD patients, potentially targeting the JAK pathway that may implicate the IL-6 pathway as well as the IL-1 family. And hopefully with deeper molecular phenotyping, this could be an inspiration for new molecular targets. So finally, I want to close by acknowledging my close collaborators at Genentech, as we can see here, as well as those from the University Medical Center Groningen, as well as the University of Leicester, and as well as Therapeutic Frontiers, who conducted the COPD rhinovirus challenge with us. And I want to thank you for your attention. And finally, the word of the day is Roche. And right now, I'll take any questions that you might have.
Well, thank you very much. That was a lot of data for us to assimilate. I must admit, I'd like you to say a little bit more about the dysbiosis of organisms. I'm not sure I understood adequately that part of the equation, how that was done technically, and how it fits into the overall hypothesis.
Yeah, so we've been studying the lung microbiome for a while now. And one thing to note that as compared to, say, for instance, the gut microbiome, the lung microbiome is a virtual microbiome desert. So the number of organisms that we see are much less than we see in other organs. However, we don't think that this is an innocuous feature in the context of disease. There has long been associations that the lung microbiome is altered in chronic lung diseases. And this is something that we observed before, but however, these had just been correlations in the past. And so one of the techniques that we, as well as other employs, is to do DNA sequencing of lung samples. And in this case, we can either be sputum or lung epithelial brushings from bronchoscopy. And from there, we're able to use PCR to amplify the 16S ribosomal RNA region in bacteria. And based on the sequencing of this region, we can identify to a pretty well degree the bacterial organisms in the airway based on our reference databases. And we can distinguish this from the host because we're doing specifically 16S ribosomal RNA, which is a feature of bacteria in the airway. So essentially, it's a sequencing technology. There are other new sequencing technologies that are improving some of the limitations of current methods. And for instance, that synthetic long read sequencing is one way that you can get down to strain identification, which is one of the limitations of traditional 16S sequencing. Did that answer your question?
Well, it answered the technical part of how you're identifying it. I think maybe I'm the only one missing the point here, but the association, once you've identified lung organism dysbiosis, how it plays into the inflammatory response.
Yeah, so at this point, we don't know that, is it a cause or a consequence? But right now we do see a correlation that, and I think to some extent this is intuitive correlation. So that subjects that have poor homeostatic control of microorganisms in the lung, they have what I really consider the smoldering innate immunity that they're unable to resolve, and it may participate as in terms of the clinical manifestations in the airway. So we don't know if the lung dysbiosis came first or a maladaptive inflammatory response. But we think that these are two features that may be contributing to poor outcomes in disease.
Okay. Others with questions, or we've got additional time for questions from our audience? Now, you attributed the neutrophilic inflammatory response to innate signaling. Again, back to the bacterial question, is it possible that the dysbiosis and things like F-met-Lu-Phe, a bacterial neutrophilic stimulator, could be involved in neutrophil accumulation?
Yeah, I think that's absolutely a possibility. One of the things I really didn't highlight is that one of the potential contributors is that that we think that may be attributed to this phenotype is that the epithelial barrier is compromised. We definitely see a molecular signature of remodeling of the airway, and we think that the physical ability to maintain a barrier is impaired, and that could be a reason why there's a greater susceptibility to colonization of the airway. And you're absolutely right. The lung microbiome could be contributing based on either their metabolites or quorum sensing molecules to actually the host response to an upregulation of neutrophilic inflammation.
All right, again, any other questions from our audience?
Well, I really appreciate all of your time and the opportunity to share some of our work.
One last question. You use the word smoke in what I would think is a very general way. I mean, we're talking particularly about tobacco incineration, or are there certain molecules in smoke that are the stimulators of innate pathways?
Yeah, that's a really great question. And as you might appreciate, and I'm sure you do, is that smoke in a very general term is not really understood what are the exact mediators that are involved. But in general, the way that I think about it is that exposure to smoke or biomass fuel exposures really results in this chronic micro injuries in the lung that through a lifetime exposure leads to the pathology that we see in the context of COPD. But as in terms of the exact mediators, I mean, nicotine has been described to have functions on immune cells as well as epithelial cells as well. And as you can appreciate, there's other toxins that are associated in the context of smoke. But to my understanding, the details for the exact mediators and how they may potentially synergize to contribute to disease is unclear.
All right, well, thank you for joining us. Unless we have any other questions or comments. we can close this session and maybe you can extend your work for us a year from now.
Well, thanks again for having me. Everyone have a great day.
All right. Bye-bye.
Bye now.